get into this. I'd like to begin by acknowledging my group, the support they provided towards this research, acknowledge my funding sources. I'm going to take 30 seconds because I have a soapbox. For those of you who are able to make admissions decisions, graduate programs, are making decisions about who might join your group, it is very likely over the next, perhaps tomorrow, several weeks, there'll be Supreme Court decisions about things like affirmative action. I urge you to be thinking about bringing people into your programs and into your groups based off of promise, based off of potential, interest in the field, not solely prior achievements. We are educators after all. Second one is that, particularly for some of you that will be returning to specific states, please ensure that your group is a welcoming environment, refuge. Not everywhere is equally friendly right now for all of the students that are in our groups. Get on the soapbox. Okay, for acknowledgement, I'd like to acknowledge this incredibly embarrassing photo of your So this is the React shared facility at Northwestern. It is managed by Schweitzer and Soled. This is a shared facility at Northwestern where a lot of the research actually occurs. So many of the facilities that you all might have in your individual laboratories you instead share at Northwestern. It enables expertise to be spread across the department, and it helps with training of people that go on to have wonderful, amazing careers. And we really should update this photo. Along those lines, I'll also do a plug. So Neil has been heavily involved in this report on rigor and reproducibility analysis. I will point you to the website, analysisrr.org, addressing questions about some best practices, recommendations for data reporting. On the experimental side of heterogeneous analysis, I urge you all to take a look. There's a formal report associated with the NSF. What do I like to do? I like to make materials. This slide I always throw at the beginning. I make stuff. There's going to be a lot of cartoons in here. This is all synthesis. I don't do computation. I collaborate with people on computational methods. I make materials that are hybrid, supported oxides, and these kind of nanostructured materials. Most of the things I'm going to talk about today are supported oxides, and we'll kind of see where all this goes. I'm also frequently asked, what do? I do everything. My group is reaction agnostic. I've been told the better way to phrase it is that we're interested in all parts of the chemical value chain, starting from the first reactions we might do, starting from chemical compounds we pull out of the ground, into more functionalized monomers and oxygenase, everything. The reason why I mention this in part is because there's a lot of talk right now about moving from an infrastructure that's based around, say, petroleum, natural gas, and other hydrocarbons to things like upcycled plastic waste, to bio-based precursors, to CO2 that you might pull out of the air. But regardless of what the initial feedstock material is, there's going to continue to be a need for new and redesigned catalysts across this whole value chain. And it's going to be for a variety of reasons. We're going to be changing our most basic inputs. It may be that certain things that are present now as waste side products are not going to become available. We need to think about new routes. Or we're going to have to start to think about small contaminants that we might have dealt with. Last but not least is that there's increasing interest in moving to distributed models where you might be doing waste transformation at a distributed site, which might mean we design more for catalyst robustness or tolerance to poisons than we do for activity. A couple of examples of this going on in the group. We've been engaged recently in a number of projects where we have a heterogeneous catalyst that is downstream of a catalyst or a chemical catalyst. So this is an example actually from a number of years ago, back in 2018, but it's fairly representative of several of the projects we work on right now. Where in this case, this was actually a bioelectrochemical cell where synthetic wastewater was consumed to generate peroxide on the cathode side. And then we were using the hydrogen peroxide generated in the catholyte to directly do a selective oxidation. And in this case, we found that supported niobium silica material was the optimal catalytic material, not necessarily because of its overall activity, but because of tolerance to water and the large amounts of phosphate salts that are present in the buffers. So it's a very different optimization scheme. And in other areas, we're doing this with, again, downstream of other electrochemical systems, downstream of photocatalytic systems, and downstream of biological wastewater treatment. At a really high level, the downstream of the biological wastewater treatment, I'm not putting any details here except to say that we're looking at propane oxidative dehydrogenation using biogenically derived nitrous oxide. I find this to be a really entertaining system. This is literally pee, where we are decomposing this in industrial wastewater systems, and they happen to generate nitrous oxide as an undesired side. You can either try to deliberately decompose this, or you can attempt to use it for productive purposes. I frequently get the argument that N2O would be far too expensive as an oxidant, but this is the case where we're presented with this, have to do something with it anyway. The primary questions actually show up here on the bottom, not so much activity, but lifetime and resistance to water. You would normally not be adding water to this system, but we are dealing with N2O that is literally bubbling out of pools. We would like to do this with as minimal treatment as possible. And so we start asking different questions. What might be poisons coming from the biological treatment of these materials? Various volatile organic carbons, which you would never encounter in propane. And how do we start thinking about this for operation and unintended use in distributed environments and by unskilled operators? Not everyone was lucky enough in this type of envisioned scenario to have a whole fleet of trained chemical engineers to be running these processes. So it's a set of interesting questions. Some of the ways that we try to address these is by building new materials. So one of the areas we started to work in is how can we take essentially process engineering solutions. In this case, this is an oxidative step embedded in a dehydro step, so specifically for ethylbenzene to styrene, where you might do a dehydrogenation and then a selective hydrogen combustion. One of the questions we've been asking is, can we build these core shell metal oxide things so we can do this on a single catalyst surface, rather than by a series of catalysts, changing from a complex process to a complex catalyst. And this has roles in things like artificial SMSIs, that JFIS chem paper that I mentioned there at the top, or this propane VH light system. We've also been involved in trying to develop zeolite-like materials, so materials that are confined at the active site without requiring extended pores that might have challenges with transport, pore collapse under harsh environments, again, with the desire to make materials more robust. So we've learned how to do things like shape-selective and size-selective oxidation on the far right. We've learned how to template chiral pores on the upper, far left. On the upper right is this templating with chiral pores. That's the JFIS MC paper in 2022 most recently. And in 2020, we reported on the ability to alter the heat of absorption of limonene at an epoxidation site. It's just a standard titanium silica type catalyst that we further modified with additional oxide to alter heats of absorption. Again, all of the types of tricks that we've heard about extensively at this meeting about altering confinement effects around the active site, almost overwhelmingly via zeolite, but trying to do this without extension. Okay, so in this talk, to actually start getting a little bit at the topic, I'm all going to be talking about oxide catalysis, I should say, and stretching the definition of oxidation catalysis a smidgen in the name of creating an interesting, to me, narrative. And so I very briefly, how do we as a group learn about supported oxide structures? Reminding you that the supported oxide catalyst structures may exist as isolated sites, small polymers, ligamers, crystallites, what have you. We can characterize them using my language. I'd say chemistry first techniques, so NMR, X-ray techniques, vibrational spectroscopies. what I call spatial techniques to give us a little bit about what is the longer range structure of these. This might be X-ray scattering, species absorption, and microscopy. I put microscopy in the longer range of things because I'm typically working with relatively light atoms that are very, very hard to visualize and support these types. And then we do probe molecule reactions and spectroscopies and electronic spectroscopies. But one of the key things I really want to emphasize is we've heard a number of good talks, even including today, about really well-defined systems and looking for kind of smoking gun effects. These systems are going to be really imperfect characters. I'm not going to have a single great way to tell you what's going on in these surfaces. So what we choose as materials is going to be really important. We're only going to see the things we're able to see because we can vary them in the way we choose our materials. So the main question is, how can we use well-defined families of catalysts to provide leads into putative active sites or mechanisms? And the leads is the key word here. And I am not going to be making very many claims about what an active site is or what a mechanism might be. But the goal for what I'm telling you in this narrative is how can we identify interesting hotspots? This would be something cool to go after. Maybe there's something going on in here. That's what I'm typically looking for. So the three vignettes I'm going to try to talk about are supported boron oxide oxidative dehydrogenation. That's closest to the actual topic here. A little bit about supported molyoxide and molysulfide catalysts, again for dehydrogenation. And a brief little bit, depending on time, on zirconium oxide-based moss for actually the MPV reaction. Okay, boron oxides. So we've heard at this meeting, certainly elsewhere, that boron-containing materials are increasingly popular for oxidation reactions. I'll point out they're proficient at initiating reactions. We heard for ODH conditions, and then there's gas. One of the challenges in trying to understand what these initiating sites might be is that bulk boron oxides can be molten under reaction conditions. And so you either need to be looking at sites that are getting generated under reaction conditions or sites transient. There's a few things that are known, and this has all come up in the last few years, that, for example, completely condensed boron in the framework is essentially inactive, whereas boron with free hydroxyl groups, labeled here as defective boron silicates, they tend to be active for initiating these reactions. And there's excellent theory, and there's excellent NMR support assignments and structural variations, but you still have many questions like that last bullet point. If you do some of the typical tricks that I would do for supported oxides, and I change the coverage of a boron oxide on a silica, you don't see a lot of surface coverage effects, which is great from the point of view that your rates for boron don't really change much, but it's bad from the point of view of trying to pick out an active site. So the proposal here is we're actually going to use the size of a molecular precursor to disperse supported borates to help identify kind of hotspots to then test against theories. And specifically, we're going to be using different sized boron precursors in what I'll call size matched silicon pores. So we're going to be putting large precursors, things like that, into pores that are 1 to 2 nanometers. My red flag here. I know this is not commercially viable. Please don't ask me about it. The cool thing about borate precursors is because of Suzuki couplings, boronic acids are incredibly available. These are the 6 smaller boric acid precursors. There's 2 more that are enormous, and they're used for appending fluorescent tags to biomolecules and things like that. So there's this huge array of synthetically accessible precursors, the main thing. Okay, we put these into small pore silica. Key thing on the left is the smaller precursors. When we put them into the small pores, they fill the small pores. We lose our smalls. On the right, when we use a wider pore silica, everything fills everything. Again, these are pre-calcination, so all the big bulky ligands are in there. The point of view is on the right, wider pores, everything goes in. Left, you have some sort of size. I've got 8 different catalysts here on the left. This is a conversion versus a selectivity versus conversion plot. Every single one of these catalysts behaves the same. In the middle there is a selectivity profile and some conversion. These will run at 500 degrees Celsius, pretty typical 2-to-1 propane to oxygen conditions. Basically, it says we've got all the same catalysts or same active sites here, just different amounts, right? They all have different rates. I'll also point out right at the front, these catalysts are stable with time on stream. There's no evidence for boron loss for these. These are low surface loadings. Boron, the borosilicates that are making are pretty stable. I'll also not get into this. There's no substantial changes in the boron structure before and after catalysis. One of the key things is we actually calcine these materials at temperatures above the reaction temperature. So any abuse that we've done with catalyst, we're doing before the reaction starts. Okay, so why is this interesting? The whole point of this was actually to develop a well-defined family of materials. So on the left, there are some NMR assignments, S1, S2, S3, S4 types of structures for boron and NMR. They mostly depend on boron framework or S3 hypothesis. And in the middle there, this is the boron, solid-state boron NMR for this whole set of materials. And I point out that in particular, they all differ in the amount of this S2 species. It's just a BO feature that has been identified as a good candidate by NMR, by NMR, and DMP, saying this is a good candidate. In the smaller core materials, the ones where some precursors get in and some don't, the content of this S2 feature varies dramatically over the whole family of materials. Whereas in wider core materials, they all have about the same distribution of boron sites on the surface. And this has also been seen as a measure. And so when you do something like boron on wider core silica, regardless of the precursor, and again, not showing, regardless of surface density, we actually see about the same turnover frequency rate per boron, which is great and all, but doesn't tell us. Instead, we use this in the smaller core silica where we got this wide distribution of boron species. We can just ask ourselves, if I plot my rate for boron against how much of this particular S2 species, I now have a putative active site. It's the only thing where rate correlates with any one of these species, S2, S3, or S1, S3. And the turnover frequency that I might get species for the more conventional supported oxides give you about the same number given the uncertainties. So again, the key thing here is that this series has identified the free BOH sites in a hydroxylated core as good candidate initiation sites for this reaction. I can strongly influence the number and the type of these sites for the deposition method precursor. And the key thing is without this family, I wouldn't have had a good experimental lead within this family to then go and do the DFT and do the additional characterization and the mechanistic sites. All right, next question, moly oxides, moly sulfides. Okay, so this is a talk about all about moly oxides. So of course, I'm going to start with moly sulfide. This is how we started the story. The key thing about moly sulfides is they exist as these platelets where the coverages on these edges, which are typically the active sites, are very dependent upon the temperature, the partial pressures of everything in here. My co-advised student, Andrew Rosen, I'm sure everyone in this room has managed to meet or tweet at this point, did a study early on, and the key thing is that he calculated the coverages of sulfur and hydrogen on these on the edges of these materials, and they depend very sensitively on reaction conditions. I will point at the second to bottom row on alkane dehydrogenation conditions. This is only to say that depending on conversion, the edges might be sulfur covered, they might be partially sulfur covered. You might not have any sulfur at all. Hydrogen, they do not. They're very sensitive little materials. It's very hard to make something, and under conditions where we might want to do dehydrogenation, just walk away and say this is. So we got into this, we were doing alcohol dehydrogenation. I should say we were not trying to do alcohol dehydrogenation, we were trying to do something else. Talk to me later. Alcohol dehydrogenation, also point out, is interesting as a method for long duration hydrogen storage using alcohols as liquid organic hydrocarbons, hydrogen carriers. I can't speak today, but it's fine, it's Thursday. We wanted to know why these things work. So the first thing we did, me being my group, is we made 4 different moly sulfide materials. I'm just labeling them as A, B, C, and D. D is a molysulfide I got from a friend. I have no idea how it was made. Molysulfides and C, I believe, was the crystalline one, and A and B were ostensibly the same material. One was made by a grad student, one was made by an undergraduate. So again, undergraduate. The key thing being that the selectivity versus conversion profiles, again, are basically the same across these materials, across different space velocities. They're basically the same materials in terms of the types of active sites they possess, but they probably differ in the type, in the number of active sites. Right. We wanted to interrogate these materials. We did X-ray absorption, we did CO absorption on the edge sites. CO absorption tells you it's noisy, these are low surface area materials, or one would be the edge sites. It tells you a little bit about what the species percentage is. I point you to 2018 catalysis reference. The key thing being that two levels of disorder, either by overall disorder from the X-ray absorption spectroscopy, or by looking at the sulfur moly edge ratio via the CO titration, tells us that the aerial rate, so the red bars on the right, tends to go up as you get more disorder on the surface. That's cool, that doesn't give me a number to work with, right? And so we wanted to ask ourselves, can we extend this? Can we quantify these findings? This is the point where, for reasons I don't fully understand, we pivoted to isobutane dehydrogenation. Yes, exactly why we did this. And the key thing here is that we can get nice, stable isobutane dehydrogenation over materials that we thought would be interesting for this because they do alcohol dehydrogenation. That's polysulfidation. Again, supported catalysts more active. We wanted to ask ourselves, are there more sites or are they more active sites? We don't have a great way to count active sites on these materials, so we wanted to set out to do that. First thing we did is our usual things. This molysulfide-R is this rag-like, high surface area, very distorted molysulfide. The -C is crystalline. This is like lubricant material, highly crystalline molysulfide planes. The rag-like, highly disordered material, both is more active from an intrinsic point of view and from presumably the number of active sites that are present. But one of the interesting things to just point out here on the screen is that the apparent activation energy for the rag-like material is very low. It's as low as the apparent activation energy you get for dehydrogenation platinum. It's several orders of magnitude slower than platinum. This is not a replacement for platinum. This whole talk should just be like labeled not commercially viable. But it has this intriguingly low barrier, which meant we wanted to go figure out what's going on. I will also mention there was a paper that had claimed that sulfide of moly oxide was as good as platinum. It's not. The cool thing about equilibrium is that once it reaches equilibrium, everything looks good. Makes it harder to publish papers. That's my butt. Okay, so we switch as we do to oxides. Why did we switch to oxides? Well, number one, Neil, who runs this lovely facility, we're here to share it with. And number two, we regained some of the characterization gaps. So we decided to look at a family of supported moly oxides. This is published in 2021. We regained our ability to do XS, to do UV Viz, to do we got some Raman, we got all sorts of things. The key thing is we made a casein-doped moly on aluminum surface. At all loadings, it's mixtures of moly oxide oligomers and aluminum lipdate because of the conditions. Surfaces are increasingly reducible as you go to higher loadings. So this is the hydrogen consumption per moly on the left. So as you go to higher surface densities, you start approaching values around 1 and then just passing a little bit. So it's more reducible, and it is also increasingly reduced. So on the right here, this actually results from an in situ X-ray absorption study where we're actually looking at an average oxidation state under reaction conditions. And the catalyst move from about +5 to about +3, depending on the surface load. I'll also mention that oxidation states under these types of conditions, under isobutane dehydrogenation conditions, are stable with time on stream. This is not shown. Any changes to the catalyst surface occur during pretreatment steps in hydrogen or essentially in the first turnover. The differences amongst these materials is that high loaded materials shown up here on the left, very selective. they're unstable. Reactivity drops quite dramatically over a few hours time on the stream, whereas on the bottom, materials that are low loading, again, quite active, but they're also quite stable. This was relatively unexpected. One of the key things is that pre-reduction was essential, and this was to move the Moly-6 as synthesized into a Moly-4 material that gets. This is contrary to what we had seen from the literature going into this, because all the literature going into this was high loading materials, which deactivate quite aggressively time on stream. As an overall view, this is again my recurring theme. We don't make a material, we make a series of materials. We look at rates, function of surface density. In green, these are our initial rates. They don't vary dramatically, going from on the far left, which is well below monolayer, to on the right, which is approaching monolayer. These are not extremely high coverage catalysts, but there's not huge variations within them. However, the reactivity after, I believe this was 8 hours time on stream, is in purple. Varies dramatically because the high loaded catalysts deactivate very aggressively. This we tried to capture in the red curve, which is a deactivation rate constant, several orders of magnitude difference. The conclusions from this, when we look at the accumulated data over the whole range of surface densities, is that all of these materials begin as Moly-6 oxides beginning. Under the initial reduction conditions, they turn into mixtures of Moly-6, Moly-4, and further reduced species that I can't really pin down. The Moly-4 species are most tightly correlated with reactivity for the dehydrogenations, as you would expect. It has coordinatively unsaturated sites due to this chemistry, and the very deeply reduced Moly sites, the ones that are correlated with coking and long-term deactivation. Deactivation is all coking. We can rebuild these materials readily. They are not restructuring time on stream. Okay, now I will tell you this all started because we got intrigued by some polysulfides for a project where we were supposed to be doing something else. We're looking at alcohols, we got alkenes, and we really asked ourselves, can we get these more closely related? Are the polysulfides and oxides more related? Can we quantitatively compare things? This data should look a whole lot like the other set of data. It's not the other set of data. You know this because it's highlighted in red. This is over-supported polysulfides, but otherwise the data looks almost identical. These materials I should mention, these are supported polysulfides by essentially a deposition route as opposed to making the oxide and then sulfiding it. So these systems were never oxidized. In this case, I overlay all of the same type of data that you saw before, whereas the oxides are the fill point and the sulfides are the buffer points. These are remarkably similar materials. I'm paranoid, so I asked if this was a coincidence. Partially a coincidence. More interestingly, we went and finally counted sites. So we started all this by saying, alcohol and alkene oxidation seems to be behaving similar. Can we at least have a marker that would connect these? So we did methanol temperature program surface reaction, detection of all the products. This gave us something we could count. Quantifying the numerous and types of surface sites, those that generate CO2, CO, ether, and formaldehyde. See, I will note that we've tried to remove most of the surface acidity from the alumina by getting this potassium. There's some questions about how much potassium is in there. The key thing is that the total sites decrease with loading. So even though you're adding more molybdenum, you're losing more sites. But you get much greater tendency towards the deep dehydrogenation, so make CO2 as you go. Again, none of this is terribly surprising. This is a nice number. And again, we would apply this correlations. We could do the apparent activation energy for forming these. And on top of your unplotting, like I did for the last slide, both oxides and sulfides. And again, it's sensitive to loading. It's not particularly sensitive to what I started with. These are definitely sulfides. No sulfides again. They can even do something wildly irresponsible, which is to look at the rate of isobutene production, cane dehydrogenation, normalized by the number of sites made from dehydrogenation. That's wildly irresponsible. But it allowed me to say, look, I've got low loaded materials and high loaded materials, and it's pretty agnostic to whether or not this is a sulfide or an oxide, which brings up this interesting notion that what's most important might be what surface it's connected to. Is it primarily in a bulk moly-moly type network, or is it on the alumina surface? And that's primarily what's dictating reactivity, not the. It also does suggest, because it enables us to actually link together bulk moly sulfides and oxides along with their supported counterparts, that the bulk moly sulfide is actually exceptionally reactive. It just has like 4 sites, very, very low surface densities. I want to do one last story very quickly. I'm going to talk about moths. Okay, so I've been hanging out with moth synthesizers for a while. I make moths. You'll have to direct those questions elsewhere. We work a lot with zirconium oxide-based moths. So here's 4 UIO66, NE1000, moth weight, and U1400. They differ in not the core. So all of these are zirconium oxide-based moths. They have exactly the same metal oxide structure. You can think of these as perfect little chunks of zirconium. They're all carboxylate ligands. They just differ in how many open coordination sites you might have. On these zirconium oxide clusters, you have acid and base functionality, and this has been discussed here and elsewhere. You have acidity from the potentially open coordination sites, you've got hydroxyls, and then you can liberate some base sites and so on. These materials are sensitive to thermal pretreatments and hydrogen pretreatments. I will say, euphemistically alter the surface acidity. So if you dehydrate and then further dehydroxylate, you'll distort these materials. You'll eliminate most of the water that can be eliminated, and you'll eliminate a lot of constant acidity. It's very weak constant acidity. But you'll generate loose. You treat these in hydrogen, and you'll regenerate constant acidity by a heterolytic activation across the ZRO bond. And since moths are perfect crystals with no defects or any unexpected features, they're the perfect. I'm being really sarcastic. It's the end of the meeting. This is the notion that we had. So they're identical acid sites and distinct pores is the idea. We always start with 3 materials, UIO66, because we can make a lot of this. In principle, the zirconium oxide core is completely saturated with carboxylate ligands, and the only open coordination sites are for defects. So when we make it, we actually put in a number of defects that are comparable to those in NE1000 in terms of open coordination sites. So NE1000, the phrasing is 8 connected, which means it's got about 4 coordination sites per node, and then Mach-808 has 6. When you treat these 3 materials, you dehydrate them and you treat them in hydrogen. This is just an IR of the OH stretching region in every single material. These are identical little chunks of zirconium oxide and solely based on the organization of the benzoate linkers around these, you get completely different surfaces. That's bad from the point of view of having an ideal perfect material with linear pores. That's great from my kind of backhanded way of saying I'm going to make an even series. I'll point this out because it's been mentioned in these Paris ACS catalysis papers. This results in a 10-fold difference in the site normalized hydrogenation and olefin isomerization rates. Again, do not use these for commercial olefin isomerization. They're pretty slow catalysts and they're really bad hydrogenation. They do it, and they're very structure sensitive. So this led us into asking, and this is my last slides about the NPV reaction. We've heard about this here today. So this is the alcohol ketone hydrogen transfer. Lost my acetone there. That's the other product. Under these types of conditions, these are running very slow. Off-808, essentially 100% conversion. UIO66, 9% conversion. This totally tracks what you would expect. This has been published recently about, off-808s, all the catalysts are doing this. NE1000 is. This is completely unexpected. NE1000 has lots of open coordination sites. You would expect it would be similarly reactive. And in fact, as I mentioned, the way we make the UAO66, it has about the same number of open coordination sites as our NE1000. Why is it so bad? I will point this out, especially at low temperatures across the board for these types of reactions, you typically see reactivity scale with just the number of open coordination sites. MOF catalysis in this respect is pretty boring. You also have relatively minimal thermal distortion, so what I showed on the prior page, where we heat everything to 250 and thoroughly abuse these MOFs, that's not going on. This is immersed in solvent propanol. We heard earlier about how formates and things like that influence active sites. They're all washed away because we're running propanol. So what's really going on here? Well, we looked at a slightly different reagent. We added a torpedo group so we could look at some cis-trans isomerization. We looked at a general selectivity versus activity type of plot. And these are reminiscent of what you would see in zeolites, where you get more confinement and you start getting more of the selectivity to cis, the trans thermodynamic products. So this was yelling at us confinement effects. These pores are large. Okay, so this now gave us an excuse, finally, to go do the DFT. We should have just stopped at MOF 808, right? We would have got our good catalyst and bad catalyst, and that's all we done. MOF 808, something like this. This is early results of modeling on the zirconium node. What you can see is a very typical mechanism for MPV. If you do this on something like NE1000, it's a little hard to tell with the linkers in the way. And so although they have nominally identical sites based off of small probe molecules, if you were doing this with methanol or you were trying to do pyridine titration, they look the same. When you've got a big old transition state, it now all of a sudden notices that it's in this very confined pore environment, and it actually has to switch into a different mechanism. So linker placement gives you alternate pathways, and we would not have seen this if we just picked the best catalyst and ended it there. Okay, so to summarize, how can we use families? Well, number one, you're limited in what you can learn from a single material. I always say this, if you have one great material and you do all sorts of amazing spectroscopy and kinetics on that, any property of that material could be the key thing. Watch out for preconceived ideas about what might be active site structures. This kind of goes into the philosophy that, please remember, we are training people to be scientists, to ask questions, not just technicians. And then interesting correlations can come from these. I emphasize these are correlations. They need to be followed up with more detailed studies that so many of the people in this room are really great. And I will take any questions.
Thanks very much.
The Exodus.
We have time for one or two questions.
Please contact me after if you so choose as well.
Yeah, thank you for the nice talk. I think as you alluded to before on, and also you work with platinum and indium,
Yeah.
you know, some of these guys really restructure under reaction conditions in ways that aren't immediately obvious.
Right.
Does that keep you up at night? I know you have a lot of exhibition characterization here and how do you think of it?
Yeah, so I did fairly sloppy in this particular presentation about merging activities.
Right.
Yeah, just find it so clear. I have to right away start.
Oh, yes. Okay. I see.
There is still no overlap.
Sure. So I'll keep these with me, and then once this one stops, back to you.
Perfect. I'll find you. Okay, so we have a ways to go.
Yes.
Yeah. It's in double turned up the backside. The other day, can you push him? So you have the here, Western, Indian, so the University of Illinois, California, and the States research program is focused on using synthetic molecular to understand protein. She was inspired to pursue research relevance after an internship at the National Renewable Energy Lab when she was undergrad. She received the bachelor degree in chemistry, PhD in MIT, and since currently she is professor and judge fellows at the University. Please, thank you very much for your invitation and also the invitation. I think one thing that you'll realize during the debate, especially at this entire session, is that I would do interviews analysis and apologies. And I don't think for all analysis that you left. But it's definitely a little bit dramatic that like you have to know. So that this is the building that I work in. We really get to spend time in there. So I very much very much counsel. That's basically speaking for the time.
Let's get into this.